So I have a slight conflict of interest in this session as I'm one of the presenters as well. This is a talk that I'm giving with Bethany Hedt, who is one of my colleagues here. The original title for this talk was Surveillance for Drug Resistance in TB and HIV, Current Strategies and Innovative Approaches. But as we were putting this talk together, we realized that actually probably a more appropriate title would be, can we borrow methods from HIV drug resistance surveillance to address gaps in current TB drug resistant surveys? So the way we're going to organize this, the next few minutes, is I'm going to try to make an argument that current approaches for surveillance for drug resistant in TB don't give policymakers at the country level enough information to begin scaling up response to multi-drug resistant TB in particular. And Bethany is going to discuss some of our work trying to use some methodologies that have been used for HIV and apply them to TB. A little bit of background information. There's an entire session about tuberculosis and tuberculosis drug resistance tomorrow morning, so I'm just going to give you just the briefest of introductions. TB is, as you know, a disease of global health importance. Nearly 10 million annual cases a year, and mortality is close to 2 million, despite the fact that we have effective therapies for the majority of strains infecting patients. You can see below I've put a table showing the standard treatment for drug-sensitive TB, and this works incredibly well, except in the case of individuals who have drug-resistant and especially multi-drug-resistant forms of disease. So MDR TB, which is mostly what I'm going to be referring to in this talk today, is defined by resistance to both isoniazid and rifampin, and it should be fairly clear with the loss of these drugs that this standard treatment is likely to be relatively ineffective. This map summarizes what we know about the global distribution of multi-drug resistant TB. Prior to 1994, there was really no systematic information collected globally on how much drug resistance there was in the world. You can see we know quite a bit more now, although there's many holes in this map. The distribution of drug resistance is quite heterogeneous, and this table here displays sort of the WHO's best guess at the total incidence of MDR annually. So their latest estimate is 440,000 incident cases of MDR-TB a year. These data are arrived at through generally two different approaches. So in some countries that have the resources to do drug resistance testing on all culture-positive cases, we're getting the best account of drug resistance in that country, and about 50% of the countries reporting this information are able to do that. The other 50% of countries, and many high-burden countries, don't have resources to test every case for drug resistance. So they rely currently on several different types of population representative surveys whereby they attempt to obtain valid estimates of the proportion of cases that are drug resistant at the country level. We've made arguments in the past about why we think these estimates actually underestimate the total burden of drug resistance. But really, even using these, what we think these estimates are relatively low, this graph really shows, I think, our failure to scale up response to the epidemic of multi-drug resistant TB. So the height of these yellow bars represent the estimates of annual incidence of cases, and we can see that only a very small proportion of these cases are either detected or treated. And in this very bottom here, I don't know if you can make this out, treated in what we think are high-quality programs for multi-drug resistant TB. So it's our hope that some alternative designs of surveys and analyses might facilitate an improved response to multi-drug resistant TB. As I said, in countries that can't do continuous surveillance of all TB cases, current surveys produce only aggregated estimates of the proportion of cases that are drug resistant at the country level. And we think that there's good reason to believe that there may be underlying spatial variability both in the burden of drug-resistant TB within countries and also the mechanism by which that drug resistance is emerging. Drug resistance to TB can emerge both among patients on therapy that we heard about in the first session this morning, that's acquired drug resistance, but individuals can also, of course, be infected by strains that are already resistant. We think that documenting the spatial variability may be important for, first of all, prioritizing response to high-risk areas, and then designing appropriate interventions based on what is the dominant mechanism of drug resistance. Obviously, interventions directed at preventing acquired resistance are going to differ substantially from those that are attempting to interrupt transmitted resistance. So in the next part of the talk, we're relying heavily on data and partners in many other countries. So I just wanted to take a second to single these people out. In particular, Matteo Zignol from the WHO has been incredibly supportive of this work and has connected us with many of these partners. I'm going to talk a little bit, I'm going to give you a couple of different examples of spatial variability in the burden of TB, and I'm going to do that from two very different types of data. First, in the Republic of Moldova, between 2007 and 2010, we've had access, myself and Helen Jenkins, who is here presenting a poster, to surveillance data, that is drug sensitivity testing, drug susceptibility testing among all culture-positive TB cases. And the second source of data are programmatic data from Lima, Peru, where DST is only offered to individuals who are considered at high risk of drug resistance. So these would be patients who are either presenting for retreatment for TB, they've had treatment in the past and now have TB again, or known contacts of individuals with multi-drug resistant TB. So Moldova is a small country. It's a former Soviet republic. It's about 160 kilometers in length. There was, prior to having good enough continuous surveillance to rely on their data, they did a drug resistance survey, a population representative drug resistance survey in 2006. This is an incredible amount of drug resistance, where 19% of new cases, those are cases who had never been treated with TB before, have multi-drug resistant disease, and the majority, 51% of previously treated cases had multi-drug resistance. This is upsetting, but not surprising in this particular region of the world, but it's among the highest levels reported anywhere. The analysis that Helen has done of data from 2007 to 2010 actually shows that this problem might be getting worse. We found that 23.5% of new cases and now 61.5% of previously treated cases were MDR. And so these are the numbers that would generally be reported up from Moldova to the WHO and pasted onto maps such as I showed you before. But this is hiding a problem. This is actually, in some ways, quite similar to what Callie showed earlier, where sort of at the country level, we're seeing incredible amounts of drug resistance, but this is masking underlying heterogeneity. So I'm going to show you this in two ways. So this is the proportion drug resistant among new cases and among retreatment cases by region. These are called rayons. And we can see that there's just a huge variation. In some of these rounds, only about 3% of new cases, while in others, about 40% of new cases have multi-drug resistant TB at the time of diagnosis. We also estimated the annual incidence of MDR in each of these rounds, and you can see, again, tremendous variability from one region to another. And I would just stress that these are on, this is a very small spatial scale that we're looking at, and we're seeing tremendous differences. Helen has a poster this afternoon that I would encourage you to go by and talk to her about, because there's some additional findings and that I think are worth looking into. The second example is in Peru. So this is a much smaller spatial area. We're looking only in an area approximately 1% the size of Moldova, but has about half the population of Moldova. So it's a very densely populated area where we were able to do geocoding on all notified TB cases between 2005 and 2007. A drug-resistant survey had been done, so a population representative drug-resistant survey had been done in Peru around the same time, where they found 5% MDR among new cases and 24% MDR among previously treated cases. So again, a serious MDR problem, but not at the same levels as what we saw in Moldova. Justin Mangiarides, who is also here today, worked on this analysis. So I want to show you two things. So this is a way in which this type of programmatic data, where not all cases is testing, requires a different type of analysis to understand. So here we have a heat map of the probability of being an MDR case at any location within our study area, and we see sort of these extended hot spots. But of course, inference on this is difficult because of the selective use of drug susceptibility testing. So this map here shows all of the TB cases in the region, and the ones that are colored red are the ones that receive drug susceptibility testing. So this is the heat map that I showed you before about the notified cases of multi-drug resistant TB. And trying to adjust for what we knew were missing cases of drug-resistant TB among those cases that weren't tested, we calculated the location-specific risk of having MDR if you were a new case and if you were a retreatment case. And we saw that the risk was highest for both of these types of disease in this northeast area of our study. And this is reconciled with this heat map by understanding that actually retreatment cases are concentrated in this region. So whereas these retreatment cases on a per case basis are less likely to have drug resistance, the fact that there's so many of them means that we also end up with a hotspot of MDR in this region. So I'm going to hand this over to Bethany now to talk about what to do about drug-resistant surveillance in the face of heterogeneity.
Thanks, Ted, and thanks to everyone for inviting us to be here. I'm going to start off by trying to be a bit faithful to our original talk and just describe briefly what the strategy is, the global strategy for HIV drug-resistant surveillance, and then talk a bit about what we've tried to learn from these strategies to detect variability in MDR-TB distributions in countries. So the HIV drug-resistant strategy, I was fortunate to be involved in the first round of implementation in Malawi when I lived there in 2006 and 2007. And this strategy is a three-pronged approach. So the first part of the strategy is an early warning indicator report that monitors programmatic data such as adherence data on individual patients or documented drug stockouts to try and identify sites that are at risk for emergence of drug resistance. The second strategy is a monitoring survey, and this follows a cohort of new initiates of HIV treatment for 1 year to see whether or not drug resistance emerges under treatment pressure. And then the final strategy, which is the one that we look for towards the most when we think about changes to the MDR-TB strategy, is the HIV drug resistance threshold survey. So the goal of this strategy is to classify prevalence of HIV drug resistance in new HIV infections. And essentially, each of the classification levels will have a different programmatic policy implication. Excuse me. So the first threshold that they look at for the drug resistant strategy is less than 5% HIV drug resistant. The recommendation is to keep the current first-line therapy options. If the area is classified as high drug resistant, so greater than 15%, the recommendation is to change the first-line HIV treatment strategy. And then classification between 5 and 15% is really looked to as a holding period where governments can review the procurement strategy so that when it crosses over into the highest level of resistance, they know to modify their treatment options. So it was this classification scheme that we looked to for the problem of detecting variability in MDR-TB, particularly lot quality assurance sampling. And the reason why we looked to this strategy is, first of all, the system is relatively simple to implement, and the classifications are linked directly to action, which we think for the MDR-TB problem is an important property for the system to have. The additional reason why we thought LQAS was a useful strategy was that there's precedence in using it to identify variability and disease prevalence and also program coverage. And I was happy to reunite with Caitlin Biedron, who I worked with a few years ago, and using LQAS as a strategy to detect variability in malaria program coverage indicators in Mozambique. So I encourage you to talk to her about the usefulness of this strategy for that particular method. So before we could adapt LQAS for MDR-TB surveillance, there were some outstanding questions that we needed to address. First, there's a variety of LQAS systems that we could use for surveillance, and so the question was which system was the most appropriate. The second is what appropriate thresholds should be used, and finally, what are acceptable errors for classification in an LQAS system. In order to explore these questions, we reanalyzed data from previously collected MDR-TB studies, focusing on the new cases, and the data comes from 3 countries, Ukraine, Tanzania, and Vietnam. So I want to spend some time to talk about the methodology that we've explored with these countries. So the first question, as I mentioned, was which of the LQAS schemes would be the most useful for MDR-TB surveillance? And we focused on 3 schemes in our reanalysis. The first is a 2-way classification where at the end of the surveillance activity, you classify areas as having high or low MDR-TB prevalence. In this simplest strategy, you take a fixed sample size and you count the number of individuals with resistance and compare it to some predetermined threshold. If you see that many or more with resistance, then you classify the area as having high level of MDR-TB resistance. And if you see less than that number, then you classify the area as low. The 3-way classification system we looked at essentially results in 3 classes, as the name implies. And in this case, you have 2 thresholds that you compare it to. Again, with a fixed sample size, if you see more than the upper threshold with resistance, you classify it as high. If you see less than the lower level resistance, you classify it as low. And if you see between the 2 different cutoffs, then you classify it as moderate. The final method that we looked at was a truncated sequential sampling LQAS, and this is actually the methodology that's used in the HIV drug resistance threshold survey to obtain the 3 classes. So with the truncated sequential sampling LQAS, you actually have a flexible sample size, and your decision rules change at each stage. So if at a given stage you see above a certain threshold with resistance, am I going to be able to get this to work? If you see above a certain threshold with resistance, you classify it as having a high level of resistance. If you see below a certain level, you classify it as low level resistance. And if you see between the 2 thresholds, then you take another sample, you test it for resistance, and then you compare it against new thresholds. Now, in theory, the sequential sampling technique can continue infinitely until you obtain one of the 2 classifications. The truncated design actually goes to a fixed stopping point. In the case of the HIV drug resistance strategy, that fixed stopping point is a sample size of 47. So if after 47 samples you haven't classified as high or low levels of resistance, then you classify as having a moderate level of resistance. So those are the 3 LQAS systems that we looked at, and we focused on 2 different sets of thresholds, trying to determine what the value of given thresholds would be and whether or not one particular set of thresholds could be universally applied. So the first system we looked at was defining low MDR-TB drug resistance as 2% or less, high MDR drug resistance as 10% or more, and moderate in between those 2. And then the second system defined low drug resistance as less than 5% and high drug resistance as greater than 20%. And then finally, because this is a sampling process and anything involving sampling isn't a perfect system, we did have to specify what were acceptable levels of errors or what properties we wanted these systems to have. And essentially, it's these specifications together with the thresholds that will determine your sample size and your appropriate decision rules. So first, for areas that have low prevalence of MDR-TB, we wanted to constrain the misclassification errors, so the probability of misclassifying low prevalence areas as high, to less than 10%. For areas that had high prevalence of MDR-TB, we wanted to constrain the misclassification errors to less than 5%. And notice that we chose to lower the misclassification errors on this end because we felt like misclassifying areas with high MDR-TB as low was a more egregious error. And then for the three-way classification systems, we actually need to give it additional constraints to define the systems. And in this case, we wanted correct classification at the low, moderate, and high thresholds to be 80% or more. This table here gives you a sense of the resulting sample sizes and corresponding decision rules for each set of thresholds in the three systems that we looked at. So you can see here for the two-way classification at the lowest set of thresholds, your sample size would be 76 and your decision rule would be 4. So in implementation, you would take a sample size. If you had 4 or more with MDR-TB resistance, then you would classify that area as having high MDR-TB prevalence. And if you saw 3 or fewer, then you would classify that area as having low MDR-TB prevalence. What I want to point out here is 2 things. First of all, notice that as you move from a two-way classification system to a three-way classification system, you need a bigger sample size in order to achieve classifications. And then second of all, when you compare the 2 and 10% thresholds to the 5 and 20% thresholds within any classification system, the 2 and 10% thresholds require a bigger sample size. Again, you're trying to pick up a finer difference in your MTR-TB prevalence, and you're going to have to pay the price in additional samples. We've actually included at the end of the talk some supplementary slides, and we're happy to discuss them, and we can also share the results of our paper. But wanted to really put the main summary points here of how these systems performed. So remember, each of these, the reanalysis of these surveys are surveys from MDR-TB prevalent surveys, where already they're reporting a single point estimate with confidence intervals. But our question was, is there any variability in these countries that's really masked by a single estimate? And what we found is when we looked in Ukraine, that Ukraine, which has a relatively high prevalence of MDR-TB in new cases at 15%, that the lower thresholds, the 2-10% thresholds, didn't identify any additional variability. So if the goal is to have additional variability identified, you would have to have a higher set of thresholds to be able to pick it up. In Tanzania, which has a low MDR-TB prevalence in new cases at 1.1%, neither system detected variability. And so using an LQAS system in that context may not be the most useful currently. In Vietnam, however, which also has a low MDR-TB prevalence at 1.8% in new cases, you would expect to see something similar to Tanzania, that maybe no additional variability is detected because you're below the thresholds that you've defined. However, in Vietnam, what we found in both systems, both the 2 and 10% and 5 and 20%, and across multiple LQAS systems, one site consistently emerged as a potential problem. And we feel like this is really highlighting the benefit of using something like an LQAS system to look locally at variability. When we talked with our partners in Vietnam and at the WHO, they weren't surprised actually that this one site was emerging. Its proximity to Cambodia, where there was a lot of movement between high-risk populations, suggested that it might be a potential problem. And again, this is something that the LQAS system was able to detect that was really masked by the single point estimate and confidence interval reported in the initial survey. I also want to mention, I pointed out before the effect of the different systems on sample size. When we analyzed the data, we allowed for early stopping in the classic system. So for example, we talked about the two-way classification needed a sample size of 76 and had a decision rule of 4. We allowed to stop early if we saw the 4 cases of resistance early on in the sampling. So in Ukraine, we oftentimes passed 4 individuals with resistance quite early on in the sampling and didn't have to go to the full 76. But what we did find is the stopping early only benefited when you had high prevalence, but you were able to stop early both at high and low prevalences using this truncated sequential sampling design. So we were able to gain some insight into what we consider to be the outstanding questions for using LQAS for MDR-TB surveillance. However, there's still some discussion about what system should be used. So in terms of the question of what are appropriate thresholds, one question is, can we come up with a set of thresholds that would be used universally across all countries? For example, what they do in HIV drug resistance threshold survey. The 5 and 15% thresholds are used in every country implementing the activity. If we're able to find a single set of thresholds that works for every single country, perhaps driven by our knowledge of the epidemiology of MDR-TB, that would simplify the development of guidelines and simplify the training of different host country partners. However, what we think can emerge from this analysis is that the appropriate thresholds may be country-specific. And so if we're really trying to develop systems to optimize our ability to detect variability, we probably want to change thresholds depending on the countries that we're operating in. The second question, which was what were appropriate areas for classification, we, ahead of the reanalysis, said 10% misclassification errors for low prevalence to high and 5% on the other end. However, we want to note that the more accurate classifications need bigger sample sizes. And again, we may want countries to be able to specify what their restrictions on classification errors might be. And then finally, what LQA system is appropriate? Two-way versus three-way, having the moderate class gives you an extra category to allow for different policy decisions. However, at this stage, two-way classification, having an idea of where it's high and where it's low, may be sufficient. And then finally, static versus sequential testing. With the sequential testing, it requires that you're able to look at samples at step by step, but now having more rapid tests for MDR-TB surveillance, TB testing may allow this option to be viable. So just in summary, we feel like we've demonstrated the ability of an LQAS-based system to detect variability. And now we're going to look at this in practice to see whether or not it works in the field. We have two ongoing studies, one in Kenya where they've decided to look at lower sets of thresholds, in part based on our results from Tanzania, where MDR-TB prevalence may be so low that having lower thresholds will be the only way to detect variability. And then secondly, in KwaZulu-Natal, where they've decided, are looking at going with a 2%, 10% system, they've actually changed the level of error to 10% at both thresholds. And some pre-analysis of existing data shows that this system will be able to detect areas with higher levels of MDR-TB prevalence. So again, we just want to acknowledge that this is work with a variety of partners and specifically thank the Center for Communicable Disease Dynamics for their funding of a lot of this work. Thank you.